Hello, and a very warm welcome to everyone joining us for today's Select Science webinar titled Improved LCMS Analysis of Oligonucleotides with Next Generation Chromatography. My name is Carrie Haslam, and I will be moderating today's session. I'm delighted to introduce today's webinar speaker, Dr. Martin Gillar, Scientific Fellow at Waters R&D. Following the presentation, we'll have some time to answer your questions. If you've got any questions for our speaker, please feel free to submit these anytime during the webinar. You can submit your questions at the left of the screen. Simply hover over the purple speech bubble icon and click to type in your question. Throughout the webinar, we will also be releasing some poll questions, so please do answer these as the webinar goes along. That would be greatly appreciated. So without further delay, I'd like to hand over to Martin for today's presentation, and I would like to thank him for speaking with us today.
So good morning, everybody. My name is Martin Gillar. I work at Waters R&D, and today I'll be talking about improved LCMS analysis of oligonucleotides. On the second slide, you can see a list of applications for genomic research or nucleic acid-based therapies, and all of these applications require LCMS tools. Here I show you one example of the method that is frequently used, ion pair reverse phase chromatography. And this method de facto became a method for analysis of oligonucleotides because it is LCMS compatible. You can see the separation here. The target RNA oligonucleotide is resolved from the impurities. Selectivity is good. Efficiency is good. The reason why ion pair reverse phase chromatography is so successful among the other methods, you can see them listed here. Ion exchange is very useful. Lately, we investigated HILIC or size exclusion. Those are all excellent methods. But the ion pair reverse phase is successful because we have developed an excellent methods and solvents that are stable in high pH, the conditions that the analysis is performed. They are fully porous, hence they can be loaded with a lot of sample. They are available in sub-2 micron particles, and the selectivity and separation is really good. I list also few papers at the bottom of the slides. Those are the papers from our lab, and they explain the mode of separation, which is in part based on charge interactions with the backbone, phosphate backbone, and in part based on hydrophobicity interaction with bases. So life is good. The ion pair reverse phase method is really good, but there are still problems remaining. Here I describe a late night in the laboratory of an hypothetical analyst. If we need to separate some samples urgently, we inject the sample and there is nothing. Previously developed method doesn't work. We can inject again and there could be a different response. We also may detect high carryover. What is going on? Ultimately, the analyst may lose confidence and move on. At the bottom of the slide, I again show two papers that describe potential explanation of what's going on, and it is called non-specific adsorption. LC systems and LC columns based on metals, stainless steel, could serve as an adsorbing surfaces for oligonucleotides, and the samples may be lost due to non-specific adsorption. At this point, that leads me to poll question number 1. Do you believe you experience loss of analytes due to metallic interactions or non-specific adsorption? So if you answered yes, you believe that you are experiencing some non-specific adsorptions, it is likely because you work with some of those problematic analytes. For example, phosphopeptides are well known that they are difficult. Multiply phosphorylated peptides may get lost. There may be tailing. Nucleotides, especially ADP, ATP, triphosphates, diphosphates, they are problematic. Obviously, oligonucleotides, they have a phosphate backbone. There could be heavily sialylated glycans and glycoproteins that have problem in the analysis. And obviously, also small molecules, and I list here an example from the paper listed below. In practice, this is how it may look. On a standard column and standard system, you may inject your sample, in this case mixture, equimolar mixture, of adenosine triphosphate, diphosphate, and monophosphate, and instead of response, you may see that there is a good signal for monophosphate, diphosphate is tailing, and you may not even see ATP, adenosine triphosphate. Second example that I show here is for oligonucleotide. Here I show how it looks like when you take a fresh column out of the box, put it on a system, and the red chromatogram is a first injection. In the sample here, I have 5 oligonucleotides from 15 to 35 mer, and you can see that some of them are missing. Injection number 2, blue chromatogram, looks better, but only by injection number 3, you start to see these minor oligonucleotides that exist in this sample, and it is still not the response that you should get. After many more injections, you may arrive to the state which is so-called passivated. The column is now giving you full recovery of the sample. This is obviously an unwanted effect. So what is going on again? Why do we have these non-specific adsorptions? There are a few papers, one of them I list here, that discuss that at neutral to acidic pH, metal oxide surfaces have a charged surface, positively charged surface, and they actively adsorb and interact and chelate with analytes, especially those that are acidic. And that also happens more visibly at low ionic strength mobile phases and slightly acidic pHs or even neutral pH. And I should also mention that this is not only a question of stainless steel material surfaces. This is present for various metals, including titanium. So that's why we embarked at Waters on a research project that we investigated whether we can detect this non-specific interaction, and I selected for this study just a frit. Why the frit? Because here I list what is the surface area of porous frits. Those are the frits that we actually use to assemble the column. Every column contains 2 frits that is holding the sorbent material in place. You can see that the smaller frit, 2.1 millimeter, has a certain surface. The larger frit, to 4.6, has a much larger surface available. And those are actually comparable or higher than the entire LC system, all the tubing, injectors, switching valves, and so on. Frit is the main part that has a higher surface and offers the greatest surface for this non-specific interaction. So if you then take a column which combines 2 columns as well as the column body, and you can see that the column has a dominant contribution to this non-specific adsorption. So we took just 1 frit, placed it in a holder, left bottom corner, and on the right corner, that holder was placed between a pump and detector. I stress here that there is no column in the following experiments that I'm going to describe. It's just a frit that is exposed to the sample. So here we inject 50 injections on a frit. This is a 2.1 millimeter ID frit, and the sample is oligonucleotide. We perform an experiment that is called multiple injections in a single experimental run, acronym MISER. And you can see on the bottom chromatogram, the red chromatogram, that first 10 injections, there is almost no signal, and the signal slowly increases. And only after, let's say, 40 or 50 injections, my signal increases and levels off at the level that I would consider to be 100% recovery. By the way, we did a control experiment without a frit. All injections were the same height, and this is how we normalized and quantified the sample. So you can see that for an injection of 2.6 picomole per injection, it takes a long time before we saturate the surface of the frit. If I do this experiment, the same experiment, 50 injections with 10 picomole sample, Within first 10, maybe 20 injections, I saturate the surface, and then the signal levels off, and my recovery is 100%. So what does it tell us? It is a direct confirmation that indeed, under the conditions that we run here, oligonucleotide sample is adsorbed on a freight. And it also tells us about the effect that is termed as a sample conditioning. As you inject more sample, the freight is passivated, all the adsorption surfaces are coated with your sample, and you are getting better recovery. For your information, it takes about 40 picomole or 310 nanograms to semi-permanently passivate the single freight. And that gets me to the poll question number 2. How are you overcoming non-specific adsorption when you analyze oligonucleotides? So some of you are using this sample conditioning, and I will talk more about all the effects that are happening here. Here I show a miser experiment with the bigger freight, 4.6 millimeter freight. The red chromatogram, again, showing repetitive injections, this time 10 injections on a low mass load, double the mass load next 10 injections, and last 10 injections with a larger mass load. You can see that my recovery is 100% for each of those injections. This is the freight that we treated with an agent. I will not mention just yet what the agent is. Nevertheless, the recovery was from the first injection nearly 100%. Contrary to that, the B chromatogram, the blue one, shows what happens for 4.6 millimeter stainless steel untreated freight. First 10 injections, low mass, there is almost no recovery, and then we slowly see this effect of sample conditioning. And towards the 30th injection, I have nearly quantitative recovery of my sample. And if you look at the value 1.4 microgram total load that I need to passivate my stainless steel freight, it is proportionally larger than 2.1 millimeter freight. So column that is equipped with larger freight, obviously, you will need to passivate them with more material. There is obviously many effects that are taking place in this non-specific adsorption. One of them, I mentioned, it's an ionic interaction. So one of the effects that has an impact on the adsorption is a pH of a running mobile phase. The red line and the red chromatogram here at 4.5 pH, you can see that the adsorption is much more prominent, and also peaks are more tailing. If I move to the same mobile phase, the same concentration of mobile phase, just adjust the pH to 7, that's the yellow or orange chromatogram, you can see that the recovery improves. And when I move to pH 8.5, green chromatogram, the recovery is almost complete. This is the good news. There are conditions that you can use to alleviate the effect of non-specific adsorption. I would caution you, though, that at higher pH, the column is obviously less stable. And if you move to pH, let's say pH 9 or above, you will start to mess up with the ion pairing reverse phase mechanism. Effectively, you need, in ion pair reverse phase chromatography, you need this ion pairing agent to be charged. And if you start to discharge it with high pH, this whole mechanism stops working. We talked about this sample conditioning experiment, and you may ask yourself a question: how permanent is the sample conditioning of your metal surfaces and freights? This experiment gives us an answer. We take 4.6 millimeter freight, stainless steel, and we perform an experiment with 25 mer oligonucleotide, and after 30 injections, the freight is more or less passivated. Then, after the experiment stops, we restart it and perform it once again. You can see that compared to red chromatograms, now the recovery is much better. The freight was indeed sample conditioned, But in the next step, I take this thread that is loaded with oligonucleotide and I take it from the flow of mobile phase and I wash it with 2.8% ammonium hydroxide high pH mobile phase, just an aqueous solution. Then we reconnect the thread into the mobile phase stream and perform the experiment with loading of oligonucleotide again. The black chromatogram shows you what happens. There is again very apparent adsorption, and it's almost like the thread is fresh and unused. This talks about what happens during the acid and basic washes. So during the basic washes, we sufficiently discharged the surface of the thread and strip off the adsorbed oligonucleotide, and that speaks about how permanent this conditioning is. It is possible to regenerate the thread and remove the adsorbed sample. It is also possible to do it by an extensive washes of your thread or column with mobile phases. So on this next slide, I've shown you many different Miser experiments, and here is another one. And I include this one because it is different. In previous examples, we used always stainless steel thread. In this example, I show you how it looks like with titanium thread. On the upper chromatogram, you can see 5 injections in 3 series of oligonucleotide. You can see that there is a familiar pattern of buildup of material. Sample was adsorbed and conditionally passivates the thread, and the recovery gets better. If you look at the bottom chromatogram, here we show different scenario for an adenosine diphosphate AMPCP molecule. It is also a molecule that is known to adsorb to metallic surfaces, and you can see that indeed we can detect adsorption in first 5 injections, and then slowly the analyte is recovered better. However, compared to oligonucleotide on the upper chromatogram, you can see that peaks are much more tailing, and you can see that during the gap between the series of 5 injections, I included those gaps deliberately, you can see that there is a tailing. While the sample was adsorbed on the thread, it is also relatively quickly desorbed by running mobile phase. If you look at the blue arrows, you can also see that the first injections in each series is actually decreased in height and area from a previous injection. That is because this sample conditioning is removed by mobile phase that desorbs the analyte. So this chromatogram neatly shows and explains why you can see tailing peaks for those sensitive analytes in chromatography, because the adsorption on the thread happens and then the material could be slowly released from the thread. It, if you think about it, also neatly explains the carryover effect. So this is my last Miser experiment that I'm going to show you. Some of you may be using sample conditioning, and some of you may be using conditioning of the entire LC system, sometimes including the column, with acids. It is known that some customers are using 30% phosphoric acid to passivate this entire LC system, including injector, all the tubings, and so on. It's quite dramatic and drastic condition. Here I show an experiment with an oligonucleotide where we took threads, put them in a solution of these shown acids, sonicated them for 15 minutes at 50 degrees C. Then we took the thread, connected it into a system, washed with mobile phase briefly, and started experiment. You can see that the chromatograms in blue, there is a good recovery. From the first injection, I have nearly complete recovery of my oligonucleotide, meaning those acids like 75 millimolar phosphoric acid or 10% citric acid actually do condition or passivate my system, my ferret, and the recovery is good. It is perhaps one can think about it as an acid treatment, but you can see that when I use on the right side 75 millimolar ammonium phosphate or ammonium citrate, the effect is similar. So it's not just an acidic interaction or acidic wash. It is thought, as we thought about it, it is believed that phosphates or citrates are strongly absorbed on the metallic surfaces semi-permanently, and they condition the system. As you can see, there are also other examples in black. 10% formic acid wash doesn't do a thing, ferrit still absorbs. 10% nitric acid, very strong acid. If anything, it actually makes the situation worse. And surprisingly, EDTA didn't have any positive effects either. Although we know that addition of EDTA improves peak shapes and perhaps recovery, but it doesn't show in this case. Okay, on the next slide, here is the summary of previously described experiments. We talked about sample conditioning. Yes, it works and you can successfully use it, but it is not a permanent solution. You can also perform LC system conditioning, including column and passivation with maybe diluted phosphoric acid or maybe with citrate. But again, it is not a permanent solution, it's only temporary. That is why we looked at more permanent solution, and there are some solutions on the market. You can use non-metallic columns, you can use PEEK tubing. Unfortunately, this is not the ideal solution. The pressure that you can use with the column is limited, and so on. So we investigated so-called high-performance surfaces. Before I get to it, briefly, we basically modify the metallic surfaces permanently and covalently, so the analytes do not have a possibility for interaction with metal oxides. The technology I'm describing here is called MaxPeak High Performance Surfaces, and it is basically conditioning or coating the entire column permanently with hybrid organic inorganic layer. That layer chemically is very similar to our hybrid silica technology that is known as a BEH, and now exists commercially under the Premier name with all sorts of Waters columns, including CSH and high strength silica HSS brands. First, we started with reverse phase chromatography. We will extend it into Hillic and other modes. So if I should now show you the difference between regular column and Acuitive Premier column, this is shown here on the slide. You have seen the left chromatogram. There is a loss of these multiply phosphorylated nucleotides. The same equimolar mixture, when it's injected on Premier column chromatogram on the right, you can see that the recovery is better. You still can detect some of the tailing, ATP and ADP. This is because we have a solution for columns, but this analysis was performed on a conventional system. So remember, the system still has some non-specific absorption, unless you treat it with conditioning. On the next slide, again, this is an example for oligonucleotides. You've seen the left panel where we demonstrate that first and second injection there is a loss of recovery of the oligonucleotide peaks. On the right side, you can see that Acuitive Premier column, the same column length, the same conditions, the same sorbent, just the Premier version of the hardware, gives you full recovery of the analytes from injection number 1. And that recovery is comparable to the fully passivated column. So that takes us to the next poll question, number 3. Would you like more information about the technologies featured in this webinar? So here on slide 22, I show you bioanalysis of oligonucleotide. And if you remember, as I earlier mentioned, that sample conditioning of a freight and metal surfaces takes longer time when we inject low mass load of a sample on the column, and it is faster with higher mass load. This is the case for bioanalysis with sensitive LCMS applications when we inject sub-picomole amount of sample. It takes really long time to condition the column, and even when fully conditioned, you may still get lower recovery of the sample than is desirable. This is shown here as a blue chromatogram. Standard acuity column, the signal is less than what we get with our Premier column. And on a Premier column, the red chromatogram, you can see about 10 times greater signal. This is reflected here on the example. The data come from Mike Donegan, my colleague, one of those days when he developed the method and prepared calibration, LCMS calibration, in an attempt to go to wider and longer study. He switched to a new column, and the signal from injection number 1, 4, 8 to 12 was lost. And it was not obvious why it is lost. We questioned whether we have a good LC conditions, whether the mass spec is still okay and has a good sensitivity. And it was only on a next slide by injection 14 and 15 when we start to see the signal for oligonucleotide. So this is the effect I'm talking about. When working with sensitive LCMS situations, the conditioning may take very long time, and even when column is partially conditioned, you may not get the same recovery and signal as you expect. That is why, by using Premier columns for bioanalysis, we achieve much better results. Here, the same separation of the same oligonucleotide at the same conditions. You can see that from injection number 1, we get a signal, and the signal very quickly levels off by, let's say, injection 6 on the proper level. Why is it still leveling, even though I tell you that the column doesn't require conditioning? It is because only the column is now treated with high Premier, high performance surfaces, but the LC system still has some metallic parts that are untreated. And even when conditioned, you may still see some partial loss of analyte. If you want to know more about how we can handle this situation, please contact water specialists. Due to time constraints, I'm not going to talk about it now. And that takes us to the next slide, poll question number 4. Do you have investment plans in this area? Finally, we arrive to the summary slide. So I believe I convinced you that oligonucleotides are a class of analytes with a strong adsorption to the metallic surfaces, and this adsorption will impact your analysis reproducibility. It may impact peak shape, carryover, calibration linearity. The adsorption problem is most apparent for these LCMS, sensitive LCMS applications, but luckily, because of the research that we performed at Waters, we developed this high-performance surface technology that now exists as a Premier columns. We have mostly columns that are targeted for reverse phase chromatography, but it is possible to perform helichromatography D surfaces, and we will extend the technology further to the next modes of chromatography later on. So now, with the proven sorbents, DEH sorbents, and well-developed ion pair reverse phase chromatography methods, when we combine these methods with high-performance surfaces technology, you will receive a Premier performance for this difficult analysis of oligonucleotides. So I thank you for your attention, and I will be happy to answer any of your questions.
Thank you very much, Martin, for that informative discussion. So it's now time for the question and answer session. So the first question we've got is, how do you know the modified engineered surface chemistry does not wear off over time?
So this is a very good question. First, the chemical modification of the surface. It's a multi-layer, cross-linked surface, and it's quite sturdy and robust. But obviously, it may wear off. For columns, the stability of those metal coatings is very good. This column is not expected to live forever. So we believe that coating lifetime is longer than the lifetime of the column itself.
Perfect, thank you, Martin. So on to the next question. So we've got, have you tested the impact of Premier columns on other categories of small or large molecules, and does the benefit accrue to these molecules as well?
Also a very good question. So this seminar was mostly about oligonucleotides as a class of analytes that adsorb to the surfaces, but there was one example, or few examples actually, with adenosine mono and di and triphosphate for small molecules that are well known to cause these adsorption problems. There are many classes of other molecules, including phosphopeptides and so on, that will benefit from those Premier surfaces. And one example that I showed using this Meiser experiment was showing clear indication that adsorption happens for small molecules, and then they also desorb relatively quickly, unlike oligonucleotides from the surfaces. So the sample conditioning for oligonucleotides actually works quite successfully. For small molecules, I wouldn't trust it all that much because relatively modest washes with mobile phase and the conditioning is actually lost. So the answer is yes, it works for small molecules. Just the small molecules behave slightly differently.
Perfect, thank you, Martin. So on to the next question. So we've got, is Waters thinking of extending the benefits of the MaxPeak high performance surface beyond the column, maybe to the metal components of the LC system too?
So this is a very good question. I alluded in my talk to the fact that, yes, while we treat the column and remove the non-specific adsorption that is happening on a column, there is still smaller portion, but there is still portion of the adsorption surfaces that are within the LC system itself. And it would be a good idea to extend the coating and the modification of metallic surfaces to the system itself. So there is an active research ongoing at Waters. We've progressed to the phase of commercialization. I can reveal that there will be a system, Premier system, with coated surfaces. It will be commercialized early next year. And if you are interested in using such a system, please let me know or write in the chat that you are interested, or contact Waters representative to have an early access to this technology.
Perfect, thank you, Martin. So on to the next question. So we've got here, what column and method would be recommended for 100 NT gRNA purity?
So I am not sure I understand the question. Could you repeat it?
Yeah, of course. So we've got here, what column and method would be recommended for 100 NT gRNA purity?
So the BEH, bridged ethyl hybrid columns that exist in a 130 angstrom pore size is generally the recommendation for oligonucleotides, and we also have a version of the same column, now with Premier surface that exists in a 300 angstrom pore size, which is equally applicable. So that would be my recommendation.
Perfect, thank you, Martin. So the next question we've got is, does this also happen to chromatography other than IPRP?
Also, very good question. I alluded in my talk that we first deploy these premier surfaces for reverse phase columns. We also looked at HILIC, and interestingly, in HILIC, those effects are perhaps even stronger. And we know that premier surfaces improve those non-specific adsorptions. I believe many users who use ion exchange columns are very familiar with this effect for oligonucleotide analysis. So it does happen in ion exchange. And I alluded to that there are some chromatographic conditions that are helpful, high pH in one example, or if you include phosphate buffers on slightly basic pH, and those phosphate buffers are 150 millimolar, well, you eliminate a great deal of those non-specific adsorptions because of the presence of the phosphate ions. Citrate buffers could also help, but otherwise, I would expect that in all modes of chromatography, under LCMS conditions, you would see those non-specific adsorptions. They are present everywhere.
Perfect, thank you, Martin. So the next question we've got is, can this column be used for mRNA analysis?
So mRNA is a much larger molecule, is apparently maybe not fitting in all those pores, but effectively with 300 angstrom pore size columns, we have done some work with large molecules, restriction fragments, DNA restriction fragments. We can successfully retain them and elute them and resolve them. So I think, yes, you could use the column, and obviously the premier version of the column would be most helpful there because the very large molecules are comparably more sensitive to these non-specific adsorptions. So yes, the premier version of these columns would be my advice.
Perfect, thank you, Martin. So another question we've got that's come through is, what do you mean with sample collection? Would you add citric acid to the sample?
That is quite an insightful question. So addition of either phosphate, citrates, or EDTA has been shown, addition to the sample, has been shown to be quite beneficial for sample recovery. We have done similar work for phosphatide. There is actually an application note that shows that EDTA really help. There is a paper from Robert Birdsall, from a colleague of mine, who shows that addition of small amount of citrate either to the sample or directly to the mobile phase could be beneficial and mask for these effects. Speaking about the recovery and masking for the effects, that is when you have a stainless steel hardware and a column. Once you have a column that is a premier version, you basically removed 80% of the problem. If now we have a QT premier system, that is, the flow paths are treated with the premier high performance surfaces, you pretty much can eliminate all those additions and modifications of the mobile phase, often not compatible with LCMS, and get really good results. So there is either solution that you will try to mask for those non-specific adsorption with metallic surfaces, or you can use the premier technology for column as well as for LCMS system and get the same effect.
Thank you, Martin. So we've got another question that's come through. So we've got, do you have to condition the system with every new oligonucleotide you test when using sample conditioning?
Good question, and the answer is no. Often we use either preconditioning with a different type of oligonucleotide that is effective and sufficiently passivate the surfaces, or it is also a good idea to include in your bioanalysis in those sensitive LCMS conditions an internal standard. It has been described by Michael Bartlett. So including an internal standard, which is an oligonucleotide, of a order of magnitude higher concentration than your sample is actually a good idea. So you continuously dynamically passivate the system every injection, and that indeed helps. So no, you don't have to use the same oligonucleotide. Actually, it's a good idea to use different oligonucleotide.
Perfect. Thank you, Martin. So another question is, thank you for the interesting presentation. What would you recommend for the analysis of a purified oligonucleotide in native conditions? I want to look at complexes between the oligonucleotide and different small ligands.
So in prep, this is not an easy answer, but in prep situations, when you overload and overwhelm the system with a sample, these effects are not as apparent as for highly sensitive LCMS application. That being said, in prep applications, the column and the Fritz are also much bigger, and they have a much bigger surface. So there might be a concern about a carryover. I must admit that we didn't look into this issue just yet. Most of our columns, or actually all of our columns that do exist in Premier are analytical scale columns. But if there is an interest in prep columns, we may extend the technology to the prep columns in the future.
Perfect. Thank you, Martin. So the next question we've got is the Fritz surface also coated in the Premier column series?
Indeed, it is, because, as I mentioned, Fritz are actually the main culprit, and these non-specific adsorption Fritz offer greatest surface in the LC system. So yes, the Fritz are coated.
Perfect, thank you. So another question we've got is there a difference in the results with high temperatures for the column, such as 60 degrees or more?
So oligonucleotide separations in so-called denaturing conditions are typically performed at elevated temperatures, often at 60 degrees C, in some cases even higher. So the Premier surfaces, the high performance surfaces, and the chemistry has to be resistant and stable in those elevated temperatures, and all the tests and evaluations that we did indicate that it is stable under low and high pH conditions. So you are free to explore separations even at, let's say, 70, 80, 90 degrees C. It should be fine.
Perfect, thank you. So another question we've got is, do you see any additional peaks in the mass spectrum due to column bleeding?
So no, if there is a peak that would indicate that the bleed related to more surface modification that is released from the column is actually concentrated on the stationary phase and alludes a certain point of the gradient. This is not happening. The bleed, there is always a bleed for any type of modification of column or the surfaces. Often they are not detectable by UV or mass spec. In this case, no, we don't see any apparent bleed or anything that would suppress our oligonucleotide signals.
Perfect, thank you. So another question we've got is, does temperature affect the passivation with acids or with the premium coating?
I want to say yes, and that's why I did the experiment at 50 degrees C. I have only anecdotal results at a different temperature, and it doesn't seem to be as efficient at lower temperature. But the jury is still out. There is no conclusive result. I have no good answer for this question.
Okay, thank you, Martin. So another question is, can the Premier BEH OST columns be used to separate modified oligos with PEG PEG versus non-modified with same length chain?
So this is a question within a question. So perhaps the answer is yes. You can use it for all sorts of oligonucleotides with all sorts of tags, including PEG. The question was probably, is there any interaction of any tags with this coated metallic surface? And so far we haven't been able to detect any. So the answer is still the same. You can use it for all sorts of analytes.
That's great. Thank you, Martin. So that's all we've got time for today. So thank you to our speaker, Martin, for today's informative webinar and discussion. And thank you to everyone joining us online and for sending in your questions. So if we didn't have time to answer your questions, we will follow up with these after today's event. If you do have any other questions, please feel free to submit them now using the Submit a Questions tab to the left of your screen, or email me at editor@selectscience.net and I will follow up with your questions. You can download any related documents in the Related Resources tab to the left of your screen, including the certificate of attendance. So if you would like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in a few days. Goodbye and thank you once again for joining us.